Roughly 15% of the people alive today carry a blood type that, by every standard rule of evolution, should have disappeared thousands of years ago. It hasn't. About 1 in 7 humans walks around with Rh-negative blood, a clean inherited absence of a single protein from the surface of red blood cells. The trait makes pregnancy dangerous, complicates transfusions, and shows up across the planet in patterns that no simple migration model can explain. For decades, the honest answer to where it came from was, "We don't know." Recent work in ancient DNA has changed that answer. The story starts in 1940 in a lab in New York. Carl Landsteiner and Alexander Wiener were working with rhesus macaque blood, injecting it into rabbits, and watching the immune response. When they brought the resulting antibodies back to human samples, most reacted as expected. A small percentage did not. The protein the antibodies were looking for wasn't there. They named the missing factor after the animal it came from. People who carried it were Rh positive. People who didn't were Rh negative. The medical consequences appeared quickly. When an Rh negative mother carries an Rh positive fetus, her immune system can treat the baby's blood as foreign and attack it. The resulting condition, hemolytic disease of the newborn, caused stillbirths, brain damage, and infant death on a scale that had gone unexplained for generations. Before the immunoglobulin treatment RhoGAM became available in 1968, tens of thousands of babies a year died from a conflict written into their mother's genetics, a quiet deletion with a body count. Long before anyone understood the biology, European nobles were already circling the trait without naming it. They called it blue blood. The phrase came from medieval Spain, where pale, aristocratic skin showed bluish veins close to the surface, a look the ruling class read as purity. To modern geneticists, the question is more direct. Were dynastic families, through centuries of restricted marriage, concentrating rare blood traits along with royal titles? Studies of Egyptian royal mummies have confirmed heavy inbreeding within ruling lines. The Habsburgs, the Tudors, and the Bourbons tightened their own bloodlines across generations, partly by design, partly by the small pool of eligible partners available at their level. The same close marriage that may have preserved unusual blood features eventually hollowed those dynasties out. Hemophilia, jaw deformities, and inherited illness cascaded down royal lines that had grown too narrow to hold themselves up. Divine right may have rested, at some quiet level, on a piece of biology no one at the time could see. When geneticists mapped Rh-negative blood across the planet, the pattern they expected didn't show up. If the trait had spread through ordinary migration, frequencies should track the routes humans took out of Africa and across Eurasia. They don't. In Western Europe, Rh-negative runs between 15 and 30%, with sharp spikes in Ireland, Scotland, and especially the Basque Country, where rates climb past 35% and no other population comes close. In North Africa, Berber communities sit between 10 and 20%. Certain Jewish and Arab populations in Western Asia reach around 25%. Then the map breaks. East of the Urals, the numbers fall off a cliff. In China and Japan, Rh-negative drops below 1%. Across most of sub-Saharan Africa, it sits under 1% as well, in some regions below 0.3%. Then come the points that don't fit a clean story. Among the Blackfoot and Blood tribes of North America, Rh-negative climbs back up toward 10%. These are populations whose ancestors, by the standard model, came into the Americas from the very part of northeastern Asia where the trait is almost absent. The genetics of the journey contradict the genetics of the destination. Researchers increasingly point to one shared feature of the high-frequency regions. They trace coastlines. From North Africa, through Iberia, along the Atlantic edge of Europe, into the Hebrides, into scattered pockets of the Americas. The pattern runs along the sea. That geography points to a specific moment in prehistory. At the peak of the last ice age, sea levels sat more than 400 feet lower than they do today. Vast, fertile plains lay exposed where water now covers everything. Doggerland in the North Sea, Sundaland in Southeast Asia, Beringia in the far north. Those plains absorbed human populations for tens of thousands of years. When the ice retreated, the seas came back. Coastlines redrew themselves inland, and the communities living on the old shores climbed, retreated, or were absorbed into inland groups. The places where Rh-negative blood runs highest today may be the places survivors reached when the water rose. The other reason the trait should have vanished is the simplest. Natural selection doesn't keep mutations that kill infants before they reproduce. Hemolytic disease killed reliably, generation after generation, in every population that carried the trait. Some countervailing benefit had to exist, or the math doesn't work. Researchers have proposed one. A parasite called Toxoplasma gondii infects roughly a third of the global population today, crosses into the brain, and slows reaction time. For a Paleolithic hunter, that neurological drag could be fatal. Preliminary research suggests Rh-negative individuals respond differently to infection, with nervous systems appearing measurably less affected. During the Black Death, between 1346 and 1353, survival rates in regions with elevated Rh-negative frequencies ran higher than in surrounding areas. The numbers aren't clean enough to declare a single genetic cause, but the correlation across two very different diseases is hard to ignore. Selection may have kept this trait alive because, in the right circumstances, it kept its carriers alive. Ancient DNA now gives the mutation a date. The RHD deletion appeared sometime between roughly 25,000 and 35,000 years ago, squarely inside the last ice age. A single gene in a small, isolated population switching off under brutal selective pressure in Ice Age refuges across Europe, North Africa, and the Caucasus. Not engineered, inherited from visitors, a deletion that survived because it cost something and paid something back. The trait is not a museum piece. Roughly 7% of the global population carries O-negative blood, the universal donor type that emergency rooms depend on when there is no time to test. Trauma wards, battlefield medics, and disaster response teams run on O-negative reserves. The Red Cross regularly warns our days from shortage. The World Health Organization estimates that around 100,000 newborns still die every year from untreated Rh incompatibility in regions where RhoGAM remains hard to get. A treatment has existed since 1968. The deaths continue regardless. Research out of the COVID-19 pandemic has suggested Rh-negative individuals may have carried a modestly lower risk of severe illness, an echo of the same disease pressure that may have pulled their ancestors through the Black Death. If this blood type runs in your family, share the group in the comments. Like, subscribe, and stay here.